Thank you. Good afternoon, everybody. Presumably, that's how it goes. Maybe it goes that way. We are a public company, so don't believe anything I say today, or don't rely on it when you buy our stock. Argos is focused on truly individualized immunotherapy based on our Arcelis immunotherapy platform, which uses patient cells programmed with messenger RNA from the patient's tumor, capturing all of the antigens unique to that tumor, and induces a unique T cell response, which correlates with all the relevant clinical outcomes. And I'll show you that data. We are focused initially on advanced kidney cancer. We've completed enrollment in a phase 3 trial. We have 2 interim data readouts next year and final data in the first part of 2017. And based on our phase 2 data, we're very confident in our chances of success in that study. The platform technology can be applied to virtually any tumor type and also to infectious diseases. We are starting trials in a number of different solid tumors, and we have an HIV program that's ongoing. And the beauty of the platform technology is that we have very little toxicity, so we can combine with active approved therapies like we're doing in advanced kidney cancer. And of course, if you're going to have an individualized immunotherapy, you better have a cost-effective, scalable way to do that. And we have built that out, and I'll show you that. So just taking a quick back step to why immuno-oncology and why people are excited about it. So the first thing you have to do is recognize something in the tumor, something unique in the tumor, and target it. And obviously, in cancer patients, the immune system is not able to do that effectively. Once you're able to do that, though, then you have to activate the immune system. Again, the tumor is avoiding recognition, but also suppressing the immune system response. And once you can do those two things, then ideally you're able to generate T cell attack against the tumor itself. So looking back at the history of immuno-oncology, we started with general immune stimulation, right? Cytokine therapy was generally just trying to overcome immune suppression, stimulate the immune system response. We then went down 2 paths, or are going down 2 paths. One addressed at overcoming the immune suppression, and the other trying to do something to recognize what's unique in the tumor. And obviously the leading approaches today, checkpoint inhibitors, which address a specific checkpoint that's limiting the immune response of existing T cells. We all know what those are. And then either CAR-Ts, which are doing something to attack a specific antigen that's found on a specific cell associated with a specific type of cancer, or the defined antigen approaches, which looked at overexpressed but non-mutated tumor antigens. So the gaps that still remain, we believe, are 1, the checkpoint inhibitors are really only addressing one specific immune suppression target, and 2, the tumor, we believe the key to attacking the tumor is identifying those neoantigens and targeting those neoantigens. And that's exactly what our therapy does, both of those, and I'll show you how we do that. Before I do, the reason why we believe attacking neoantigens is the key is that when you look at the variety of mutations found in solid tumors, they're not in common. There are not very many in common from patient to patient. So for example, in a breast cancer study that was published a couple of years ago, they looked at tumors from 50 different patients, found 1,700 different mutations. Only 3 were found in common in 5 or more of those patients. And a study that just came out of University of Chicago earlier this week found millions of mutations just within the same tumor from one patient. So what we do to address capturing those unique neoantigens for the patient is we take messenger RNA directly from the patient's tumor. We need a very tiny amount of tumor, and we capture all those unique neoantigens for that patient's tumor. To address the immune suppression, we've identified a breakdown in immune response attributed to a lack of signaling from CD4 cells to dendritic cells, and that signal is CD40 ligand. So we provide the CD40 ligand along with the messenger RNA into the dendritic cell that we make from the patient's cells. And the result is we get copious amounts of IL-12 production, which then turns naive CD8 T cells into memory T cells, which stimulate killer T cells. And those killer T cells are specific just for the tumor because the dendritic cell is expressing those antigens that are unique to the patient's tumor. We have a very effective manufacturing process. As I said, we need just a tiny amount of tumor that comes from the patient's tumor. In the case of kidney cancer, these patients have surgery. It's stable at room temperature. It's shipped to us. We have up to 10 days to receive it. That provides the messenger RNA. And then we collect a leukapheresis, like many cell therapy companies. We have up to 4 days to receive that at our centralized manufacturing facility. And from those two autologous components, we're able to generate on average 16 doses of product, and it's frozen and shipped to the site on an as-needed basis, dose by dose, and stable for up to 2 weeks while frozen during the shipping. So why kidney cancer? So the first reason is that we believe that kidney cancer tumors are immunoresponsive. High-dose IL-2 is still really the only thing that cures advanced kidney cancer. And we're able to combine with the current standard of care, which is tyrosine kinase inhibitors or VEGF inhibitors, without adding any additional toxicity. Currently, kidney cancer is treated with sequential monotherapy. There's no combination available. So we're able to come in right on top of current standard of care. We're dealing initially with patients who are metastatic at the time of diagnosis. And when you look at kidney cancer, it's actually highly predictive in terms of overall survival at baseline based on a risk factor model that has been validated from a large thousands and thousands of patients. And the risk factor model predicts survival, as you can see, with very tight windows. And these patients who we're treating are intermediate and poor risk patients, and in particular are in the first group have that first risk factor, every single one, from diagnosis to treatment of less than a year, because when they walk in the door, they have metastatic disease at the time of diagnosis, as opposed to favorable risk patients who have no risk factors, who were diagnosed in a prior year, eventually become metastatic, and only then are treated with TKI at that point in time. So when you look at the specific patient population who comes in the door with that first risk factor, this is from the same group database, the survival of those patients with one or more of those risk factors, but specifically that one, is 15 months. And when you look at our phase 2 data, where we treated a similar patient population, our median overall survival was 30 months, with only 50% intermediate risk patients and 50% poor risk patients, so a more difficult patient population. And not only did we double the expected survival, but we also doubled the expected progression-free survival and had a much higher than expected PR rate. Probably most importantly, though, is the correlation that we saw between survival and immune response. So in the 14 patients who received at least 5 doses, you can see that other than really those 2 outlier patients, there was a direct correlation between the magnitude of the T cell response, and these are tumor-specific T cells generated by our product, which actually are PD-1 negative, so they're negative for PD-1, not subject to checkpoint inhibition, and it's the absolute increase above baseline. So you have to take the total number of these T cells after 5 doses and subtract what was there at baseline. So there are pre-existing T cells that are tumor-specific, existing at baseline, but they don't correlate with any clinical outcome. Perhaps those are PD-1 positive, something we're looking at in phase 3. But when you look at the absolute increase in these tumor-specific T cells induced by our therapy, there's a direct correlation, and this correlation exists not just with survival, but also with PFS and the amount of tumor regression. So this led us to move into our phase 3 trial, basically the same design as phase 2. I'll show you the design in a second, with a couple of important enhancements. First is that this is an overall survival endpoint, and we're looking at a median overall survival in the experimental arm versus control, which based on our SPA is about a 6-month improvement in survival. We're using 16 months as our control arm assumption, so we're being a little bit conservative there. In particular, we saw in phase 2 that the intermediate risk patients had a median survival of 62 months, which is about 3 times longer than expected with standard of care. So we designed the study to favor the inclusion of intermediate risk patients, and it worked out exactly the way we wanted. So with all patients now enrolled, approximately 73% are intermediate risk, meaning 1 or 2 of those risk factors, and the remainder are poor risk. But only patients with 3 or 4 risk factors with the poor risk group. So we eliminated the poorest of the poor risk patients. We want patients who will be on study for at least 6 months to receive the first 5 doses. That's the point in time where that immune response correlates with expected clinical outcome. So we wanted to make sure the vast majority of patients are getting at least that 5-dose induction phase. And also, unlike in phase 2, we're dosing through progression. So in phase 2, patients, as soon as they progressed, came off study, didn't receive any more AGS-003. In phase 3, they stay on AGS-003 through at least 1 progression. Of course, to do that, we allow the patient to switch off Sutent. So the design is 2-to-1 randomized. We enrolled 460 patients, 462. Two-thirds of the patients start on Sutent with AGS-003, but then they can switch from Sutent either for toxicity or progression at any point in time after the first cycle of Sutent, continuing on AGS-003. Control arm, Sutent, followed by standard of care, targeted therapy afterwards. As I said, the trial is fully enrolled. We enrolled it over 130 global sites as far as Israel and Europe, all shipping back and forth to our one centralized facility in Durham, North Carolina, without any problem. We have a well over 90% manufacturing success rate and delivery of doses. And of the patients who could have received their 5th dose, at this point in time, over 90% of the patients have received their 5th dose. So we think the trial is going about as good as we could have expected. And as I said, we have interim data coming up next year at 50% of events, 75% of events, and 290 events are needed for the full statistical analysis. We expect that to happen in the first part of 2017. So very quickly on the commercial manufacturing business model, it's quite different from what you've seen from approved cell therapies. We only need 1 cell collection. We have up to 10 days or more to get the product to the patient because it's frozen and it's shipped in a frozen shipper that keeps it at target temperature for at least 10 days. All they have to do is take it out of the shipper 30 minutes before intradermal injection and then inject it, and that's it. Our dosing schedule is 5 doses, 3 weeks apart, and then we go at week 24 to the 6th dose, and then it's every 12 weeks for boosters, so 8 doses in the first year. And as I said, from our 1 production run, we can get 16 doses, which is 3 years of dosing. And for our long-term survivors, they just come in for a second leukapheresis. We use the same RNA, and we keep dosing. And we are moving to automation, which will support our scale-up and commercial launch. We've developed automated devices that do the whole manufacturing process start to finish. We're building a commercial facility, which will be ready next year. We'll have the ability to have manual capability at that facility, as well as automation, and transitioning to full automation as we need to scale. So that facility will be able to support 10,000 patients at peak. So we'll be able to support the US market and Europe, at least for the first few years as well. As I said, it is a platform technology, and so we're able to move into other tumor types without any development work at all. We have an early-stage RCC trial that's ongoing. We're starting a non-small cell lung cancer study later this year. A bladder cancer study is being done at Mayo Clinic starting early next year, and a study in pediatric GBM also will be starting early next year. So it's the exact same process, everything identical to what we're doing in kidney cancer, so easily transition to other indications, combining with other therapies, or when we go early stage, like in kidney cancer, it'll be a standalone therapy. So in conclusion, we have that platform technology that induces that unique T-cell response. We're in phase 3, and we expect data over the next 18 months with final data. We can apply this technology across the board to virtually any tumor type, as well as our HIV program that's funded by NIH. And we have a scalable, cost-effective commercial model. Thank you very much.